Welcome to our lecture on the luteal phase of the estrous cycle. This presentation was adopted with permission from the lecture of Dr. John J. Parrish. At the end of the lecture, students would be able to describe the endocrinology behind the luteal phase of the estrous cycle in domestic animals. The diagram shows the estrous cycle of the cow, showing the luteal phase as well as the follicular phase. For the luteal phase of the estrous cycle, it is encompasses day 0 to day 18 to 19 of the estrous cycle. As you can see here, the day 0 represents the ovulation. After the luteal phase, we also have the follicular phase. The luteal phase is dominated by the formation of the CL or the corpus luteum, which produces the P4 or progesterone. As you can see in the diagram in the earlier phase, as the CL begins to form after ovulation, it does not produce much P4. About 5 days post-ovulation, the P4 significantly rises or increases rapidly. It peaks at diestrus until the CL undergoes luteolysis, where the P4 drops. Luteolysis is of course brought about by PGF2 alpha. After the luteal phase, the cycle goes back to the follicular phase again, and the follicles again grows and ovulate. Note that in the cattle and the sheep, we have multiple follicular waves, but only the last one will ovulate. So this diagram shows the preovulatory follicle. It is the one that will later ovulate, and it requires, of course, now the preovulatory LH surge. It has the theca interna. We also have the basement membrane and the granulosa cells. So the basement membrane is the structure that separates the theca interna from the granulosa cells. So in the preovulatory follicle, we have the steroid synthesis, particularly progesterone. So the steroid synthesis begins to increase prior to ovulation. There is also the production of the collagenase. So the collagenase is an enzyme that will break down the theca interna and also the basement membrane for ovulation. The structure that is formed after ovulation is the corpus hemorrhagicum. It is formed by the rupture of the blood vessels that are located in the follicular wall. The blood will fill the cavity, and this structure will form the corpus hemorrhagicum. This structure lasts for about 1 to 3 days after ovulation, and at this time, the walls of the old follicles collapse, the cells intermix, and the old basement membrane becomes connective tissue of the corpus luteum. Next phase is the formation of the functional CL or the corpus luteum. So as the cells intermix, the CL will increase in size. We also have the formation of the large luteal cells and the small luteal cells. So take note that the large luteal cells were derived from the granulosa cells, and the small luteal cells were derived from the theca cells. Because this is the functional CL, there is already an increase in the production of the P4, and a small cavity may also be present where the follicular antrum was present. There are two important cells that are found in the functional CL. We have the large luteal cells and the small luteal cells. The large luteal cells are again derived from the granulosa cells of the antral follicle. The small luteal cells are derived from the theca interna of the preovulatory follicle. This is a closer look of the large luteal cell and the small luteal cell. Are mitochondria that are associated with both the large luteal cells and the small luteal cells, because these structures are important for steroid production. So of course, in the synthesis of steroid hormones, we first have to convert cholesterol to pregnenolone in the mitochondria. Another structure that is found is we also have the black dots now that are the secretory granules, and this contain either oxytocin or relaxin, but not both. The relaxin is of course important in parturition, while the oxytocin is important in the regression of the corpus luteum. The large luteal cells increase in size by hypertrophy, and that is threefold. For the small luteal cells, they also undergo hyperplasia or an increase in number, fivefold, as well as some hypertrophy. The CL also has a rapid vascularization, and this is initiated by androgenic factors coming from the follicle. The vascularity is important for the steroid synthesis, particularly P4, as well as the delivery of the hormones. Failure in hypertrophy, hyperplasia, and vascularization would lead to a non-functional CL. So a non-functional CL will regress early and will fail to maintain pregnancy. The P4 or the progesterone has several actions. It has a negative effect on the hypothalamus, particularly on the kisspeptin neurons, and also in the anterior pituitary. The P4 has a positive effect on the mammary gland. It will promote the development of the alveoli. The P4 has also a positive effect on the endometrium of the uterus and a negative effect on the myometrium of the uterus, promoting quiescence to inhibit its contractions. Note that at or near parturition, P4 would go down, allowing contractions of the uterus for parturition. What controls P4 production? This diagram shows the mechanism whereby the luteinizing hormone will act on the luteal cell of the CL. So in here, we have here now the LH receptor that is located on the luteal cell. So this receptor is a GPCR or the G protein coupled receptor. The LH works by using the second messenger system, cyclic AMP, as shown in the diagram. The cyclic AMP will enhance now the PKA or the protein kinase A. It will activate histones to change DNA transcription. The new mRNAs will induce protein synthesis in the RER, and it will affect the cholesterol uptake in the mitochondria. So in the mitochondria, it is where the cholesterol will be converted to pregnenolone, and later in the smooth endoplasmic reticulum, it is where the P4 will be synthesized. The CAMP via the catalytic unit of the pKa can promote the uptake of cholesterol into the cell via the cholesterol LDL receptor. This simulates the cholesterol esterase to split the cholesterol, as shown in the diagram. Cholesterol is taken up into the mitochondria. This process is considered to be the rate-limiting step in the formation of P4. And it depends on the amount of cholesterol that is taken up into the mitochondria. The cholesterol uptake into the mitochondria is key in making P4. Both the small and the large luteal cells have LH receptors now for the production of P4. The small luteal cells have LH receptors and they work by the mechanism as shown previously. Note that P4 production is driven by LH. The small luteal cells will require LH for P4 production. For the large luteal cells, on the other hand, they have a very few LH receptors, and the P4 production is independent of LH. Luteolysis is the regression of the CL, and there are various factors that will influence luteolysis. Let us first talk about the role of uterus in luteolysis. This is a diagram of the effect of uterectomy or the removal of the uterus upon the CL and the estrous cycle in the U. So these experiments were done and we get similar effects in the cow as well as in the sows. So in the intact uterus, the CL lifespan is the same as in the normal cycle. So it is about 15 to 17 days. If we are going to remove the uterus on the opposite side, now the opposite side or the contralateral side as shown in the diagram, a normal lifespan of the CL will also be obtained. So that is about 15 to 17 days lifespan. With total uterectomy, so total uterectomy is the removal of the uterus as well as the uterine horns. The CL lifespan is similar to the gestation length, and in the U, the gestation length is about 148 days. So that will also be the lifespan of the CL, and that is longer than the usual 15 to 17 days. A partial uterectomy or the partial uterectomy that is ipsilateral to the CL will cause the CL to have a lifespan longer than the normal. So that is about more than 35 days. So the normal is 15 to 17, and we are going to if we are going to remove the CL ipsilateral or the uterus ipsilateral to the CL, the lifespan would be about 35 days. So from this experiment, we can conclude that the uterus produces a substance that will cause luteolysis. Removal of the uterus ipsilateral to the CL will cause the CL lifespan to increase, and the removal of the uterine horn contralateral, or on the opposite side of the CL, does not affect the CL. So how does the prostaglandin get now from the uterus to the ovary? So this diagram shows the control of luteolysis by PGF2 alpha. So of course we can go back now to our estrous cycle, and as we have discussed, at day 15 of the estrous cycle, the progesterone from the CL will stimulate the production of the PGF2 alpha coming from the uterine endometrium. So the prostaglandin is synthesized by the uterine endometrium, and it is released into the uterine vein. So in the cow, the prostaglandin cannot go into the bloodstream or the systemic circulation directly into towards the ovary, because there is an enzyme in the lungs that destroys the prostaglandin. As shown in the diagram, we have here our uterine vein, as well as the uterine artery. And the uterine vein is the one that will drain the ipsilateral, or the same side of the uterus. So we have here our uterine vein in blue, and it is the one that will drain the ipsilateral side of our uterus. The prostaglandin is again produced in the uterine endometrium, and it comes down to the uterine vein. It will then be picked up by the uh, uterine artery, as well as the ovarian artery, through countercurrent exchange and delivered back to the ovary, where it will cause the regression of the CL. This diagram shows the effect of uterectomy or hysterectomy in the lifespan of the CL in the mare. So the horse is considered to be different when compared to the cow, the ewe, and the sow. If we are going to have an intact animal, the normal lifespan of the CL will be maintained. If we are going to do total hysterectomy, so if we are going to remove all of the uterus as well as the uterine horn, the CL lifespan will be maintained. For a contralateral hysterectomy, so if we are going to remove the uterine horn, opposite to our CL, the CL in the left will be maintained. So there is a 50% of the CL will be maintained, and 50% of that will undergo regression. So, and if you are going to do that now on the ipsilateral side of the CL, removal of the uterine horn ipsilateral to the CL will also have a 50% of the CL will be maintained, 50% of that will be regressed. So one of the major difference now in the production of the prostaglandin in the mare is that the prostaglandins go to the bloodstream or the systemic circulation in the mare, as compared to that of the cow, the sow, and the ewe. Another major difference is that there is no winding around of the ovarian artery with that of the uterine vein, as shown in the diagram. This means that although the uterus produces prostaglandin, the CL in the mare is much sensitive to the prostaglandin than that it is not to the cow. That is the reason why 50% of the CL is still maintained even if you remove the uterus in the other side. This is because the uterus can still produce enough prostaglandin to cause the regression of the CL. This diagram shows the effect of prostaglandin on the CL relative to the number of days of the estrous cycle in the cow, the ewe, and the mare. Of course, for the days of the estrous cycle, for these animals, the estrous cycle is 21 days, and the day 0 here represents the day of estrus. The y-axis represents the progesterone levels in nanogram per ml. So in these animals, in the first 5 days of the estrous cycle in the cow, the ewe, or the mare, so after the CL forms, the CL is not responsive to prostaglandin. It is only later in the cycle that it becomes responsive to PGF2 alpha. So shown in the diagram, This portion here is the days now wherein the prostaglandin is not effective, and after that, no, the PGF2 alpha would be effective no, for these animals already. In the sow, it is even longer. So kanina, no, in the cow, the ewe, in the mare, the period wherein the PGF2 alpha is not effective is 5 days now after the CL is formed. While for the sow, it is longer and it will take about 12 days or so. The PGF2 alpha will cause the luteolysis of the pig corpus luteum only after day 12 of the estrous cycle. This diagram shows a plot of the hormonal changes during the estrous cycle, particularly of the hormonal changes in the PGF metabolite in the blood. We also have here the levels of the oxytocin as well as the P4. First, in the estrous cycle here, now we have a dominant level of P4, so that is in the diestrus. It comes down at sometime at day 16 of the estrous cycle, where the luteolysis normally occurs. We also have here a measurement of the levels of the oxytocin in the blood. And note that every time there is an increase in the PGF, it is also preceded by an increase in oxytocin. So prior to luteolysis, we can already see here an increase in the PGF to alpha, and it is also preceded here by an increase in oxytocin. The same pattern is also seen in during luteolysis. Of course, during luteolysis, there is an increase in the level of the PGF2 alpha, and we can also see here that it is preceded by an increase in the level of the oxytocin. This diagram shows the relative levels of the endometrial oxytocin receptors on the different days of the estrous cycle in the ewe. Of course, the estrous cycle in the ewe is 17 days. So looking at the uterine endometrium, note that there is already the presence of oxytocin receptors at the different days in the estrous cycle. Oxytocin, of course, will stimulate these receptors, causing the release of PGF in the uterine endometrium. So early in the cycle, as shown in the diagram, you can also already see oxytocin receptors, although the CL is not yet responsive to PGF2 alpha at this time. The oxytocin receptors are reduced mid-cycle, so shown in the diagram, and return when the PGF2 alpha is being released now in the endometrium at day 16 to 17 of the estrous cycle. The uterine endometrium begins to be sensitive to oxytocin again and release PGF2 alpha in return. Where does oxytocin come from? Oxytocin is produced in the hypothalamus and released in the posterior pituitary. Oxytocin causes the initial release of PGF to alpha as we get oxytocin in the uterine endometrium. So once we start getting more prostaglandin, we also get more oxytocin coming from the CL, particularly from the large luteal cells. These are the effects of luteolysis on the CL. The first is decrease in the blood flow. The PGF2 alpha will cause a decrease in blood flow to the CL. That is because the PGF2 alpha has a vasoconstrictor activity. The next are the apoptosis as well as the decrease in the progesterone synthesis, particularly a decrease now in the P4 synthesis. So this diagram shows the molecular mechanism of the PGF2 alpha that is important for luteolysis. So this mechanism will utilize the calcium as a second messenger. So the PGF2 alpha will bind to its receptor on the CL, and the receptor is a GPCR. So it will utilize the IP3 or the inositol triphosphate, and it will stimulate the endoplasmic reticulum to release calcium. We also have here that the AG, the acetylglycerol, and the calcium will also stimulate the calcium channel to open via the protein kinase A. So it will stimulate the calcium channels to open, causing the influx of calcium. The calcium is important to stimulate apoptosis, as well as we also have here the protein kinase C The protein kinase C is important to inhibit the formation of progesterone from cholesterol. There is also an evidence that in the process of luteolysis, the immune system is involved. The lymphocytes and the macrophages will invade the corpus luteum, and this will create a signal that will trigger apoptosis and CL regression. So what are the actions of the PGF2 alpha on the cells of the corpus luteum? For the small luteal cells, so they have a very few PGF2 alpha receptors, while the large luteal cells have a numerous PGF receptors. Although the small luteal cells have a very few receptors for PGF2 alpha, they will also trigger the lysis of these small luteal cells together with the large luteal cells. So we time the menstrual cycle by the beginning of the menses. There is also the menses at the end of the menstrual cycle. So just like in domestic animals, we also have the follicular wave, and we also have here the process of recruitment, selection, dominance, and ovulation. After that, now is the formation of the CL, and the CL will last for about 13 to 14 days now in primates. In the follicular phase, we also have here the dominant hormone is the estradiol, and that comes from the follicle. And in the diestrus, we can also see here the presence of the P4 and the estradiol, and that is a characteristic of the primates because the CL produces both the progesterone as well as the estrogen during diestrus. In humans or in primates in general, we can hysterectomize a female and it will not affect the lifespan of the CL. The lifespan of the CL in human is about 12 to 14 days. However, in the absence of pregnancy, the CL will self-destruct. The CL is absolutely dependent on luteinizing hormone as well as the human chorionic gonadotropin or HCG. The luteal cells will start to die in the absence of the luteinizing hormone. Then it will produce an intraovarian PGF2 alpha and probably oxytocin that will work locally on the CL to cause luteal regression. So during menstruation, there is a drop in the levels of the P4 as well as in the estradiol. There is also the release of the endometrial PGF2 alpha, and it will cause vasoconstriction as well as necrosis. The cramps that women experience during menses can be due to prostaglandins. There is also the endometrial inflammation as well as tissue degeneration.